Today's chair and speaker, our speaker organizer, is Dr. Tony Gutierrez, and Tony is an associate professor in environmental microbiology at Heriot-Watt University. So I'll pass you over to him. Tony, over to you.
Thank you, Angie. So it gives me great pleasure to chair this session on such an important topic. PFAS is organohalides is very important pollutants of our environment. I'm not going to go into any great introduction here. The biographies of the speakers are online as advertised, so I'm not going to go into any detail in introducing them. I think we should just get on underway for Dr. Sophie Holland to kick off the first talk. So I welcome Dr. Holland to give her talk on bioremediation of chloromethanes in anaerobic subsurface environments.
Cool, thanks so much. And thanks Angela and Louise as well for putting everything together. Let me just get my screen sharing. Screen number 2. All right, I think that is the right one. Cool. Okay, so yeah, as Tony introduced, I'm going to be talking today about bioremediation of organochlorines and specifically of dichloromethane within anaerobic subsurface environments. So this is work that I did during my PhD with my supervisors, Professor Mike Mainfield and Dr. Matthew Lee at UNSW. And yeah, thanks again for the opportunity to present. It's really nice to at least be able to come together for more international audiences in light of everything else terrible that's happened in the last year. So yeah, to start off with a very broad introduction, just to kind of get everyone on the same page. Essentially, organochlorines are problematic pollutants in the environment, and it's this big range of chlorinated hydrocarbons that include chlorinated alkanes, ethenes, ethanes, and methanes. Some of the more notorious ones you may have heard of are vinyl chloride, which is used to produce PVC, and the insecticide DDT. But today I'll be focusing in on the chlorinated methane, so I'll be speaking about dichloromethane degradation predominantly. And so essentially, when we look at organochlorines, they have a lot of valuable properties, which is why they've been used so extensively in industry, but these often make them quite recalcitrant pollutants. And so what we see when organochlorines are released into the environment is when they're typically denser than water, so they sink down through the water table here until they hit an impermeable membrane such as bedrock or clay and form what is called a dense non-aqueous phase liquid zone or DNAPL zone. We then see as groundwater flows over that zone and picks up little bits of solvent and carries it along with it, that you end up with this big dissolved plume that kind of flows away from the source site of contamination. And so this is an issue because a lot of these organochlorines are dangerous to both human and environmental health. And so when we look at bioremediation options for this, typically if we want to use microbes to remediate this kind of pollution, because we know a lot of different microbes, bacteria now that can degrade these organochlorines, you can look at natural attenuation, which is essentially when the site will kind of remediate itself. It's already got the right conditions and the right bacteria there, and we just need to monitor it over time. But there's also more interventionist approaches, so we can use biostimulation, which is the addition of nutrients to stimulate those organisms that are going to be able to degrade the pollutant, or we can go the whole way and do bioaugmentation, which is addition of the organisms themselves into the environment. So typically, when we think about anaerobic bioremediation, and that's really important for these kind of sites because often down near this DNAPL zones and the dissolved plume, it's quite low redox or anaerobic environments, so anaerobic organisms are really important for bioremediation of this kind of contamination. And typically, when we're looking at organochlorines, the pathway involves anaerobic respiration. So that's also known as reductive dechlorination. And it's basically, there's an example here, you get chloroform acting as an electron acceptor and hydrogen acting as the electron donor to dechlorinate it and produce dichloromethane. So these reductive dehalogenases that catalyze these reactions are quite well studied. And what's really important about them is that they have a cobalamin cofactor that has this centrally ligated cobalt atom. And that's a really strong nucleophile that can attack the carbon-chlorine bond. And so this cobalamin cofactor is going to be important later when I'm talking about candidate enzymes for dichloromethane dechlorination. But essentially, what's also good about these reductive dehalogenases is they're quite well studied, and we can quite easily track a lot of them in situ these days. So various environmental consulting companies, if they're looking at remediating a site, you can go in and see, what kind of levels of these reductive dehalogenases are already present, or if you're adding in nutrients or you're adding in microbes, you can track over time and hopefully see that level of reductive dehalogenases increasing. So when we come to dichloromethane, a little bit more information on that. It's been widely used as a solvent and a degreaser and a propellant amongst many other uses, but things that have made it really valuable in industries, such as the fact that it's miscible with all these different organic solvents under standard conditions, have also made it a really recalcitrant pollutant. It's still being widely produced and widely used, so about 900 gigagrams per year are produced, and 70% of that is from humans. The other 30% is actually naturally produced dichloromethane. And what we see in the environment is basically that despite also coming from natural sources, it is a big issue because it's a probable carcinogen that can also damage the central nervous system and reproductive system in humans, and it's also associated with kidney and liver toxicity. As well as this, it can cause issues for microorganisms, so it's inhibitory to a lot of different microorganisms. Organic solvents like DCM can cause damage to cell walls, as well as being inhibitory to other enzymes within the metabolic process. And it's also recently been discovered as actually a quite serious ozone depleting substance. And so that's something that's causing a lot of issues with a lot of dichloromethane that's being released atmospherically as well. So when we look at this map, which is from ToxMap, which has sadly now been discontinued since December 2019, but this was the most up-to-date version of it at that point in time. And it shows a bunch of all of the national priority list sites within the United States that contain dichloromethane, which is about 30% of the national priority list sites at that time. And those are the squares on this map. We can also see what was listed as dichloromethane releases into the environment from the 2016 toxic release inventory, and that's these variously sized circles on the map as well. So basically, DCM is quite a widespread contaminant. It's very prevalent. And while we do know quite a lot of aerobic DCM degrading bacteria, which are typically methylotrophs, as I mentioned on the previous slide, these contaminated sites are often really low redox anaerobic zones. And so anaerobic degradation of dichloromethane is really important to be able to move forward with remediation of a lot of these sites. Now, contrary to the sort of common model we have of this anaerobic respiration of chlorinated compounds, we only know 3 well-characterized dichloromethane degrading anaerobic bacteria thus far, and they actually all ferment dichloromethane. They don't respire it. And it's interesting, it is thermodynamically possible for dichloromethane to be anaerobically respired, but currently all the bacteria we've found carrying out this metabolism are actually fermenting it, and they're all bacteria that are within the Peptococcaceae family. So the 3 known dichloromethane fermenters are all marked here on this 16S phylogenetic tree, and they're pretty closely related, particularly Candidatus dichloromethanomonas, to other organochlorine respiring bacteria, such as the Dehalobacter species that can respire chloroform, chlorinated ethenes and methanes. So there's actually only a single isolate amongst these 3, which is Dehalobacterium formicoaceticum. It's the only isolate we have capable of dichloromethane fermentation, and the other 2 Candidatus species are present in enrichment cultures. So what I'm going to be speaking about today is Candidatus formomonas warrabia, or DCMF, as we've been calling it. And that is an organism that we have got present in an enrichment culture called DFE that's been enriched from a contaminated aquifer near the Botany Bay Industrial Park in Sydney, which is right near UNSW, where I was working on this. So essentially what we know at the moment is that dichloromethane is transformed via the Wood-Ljungdahl pathway. So the Wood-Ljungdahl pathway is this really ancient metabolic pathway, potentially one of the first pathways on Earth, and it's typically used by acetogenic bacteria and methanogenic archaea to produce acetate from hydrogen and carbon dioxide. Now, there appear to be variations on this pathway that the dichloromethane fermenting bacteria use, and the big question at the moment in the field is essentially, what is the enzyme that is transforming dichloromethane into most likely methylene tetrahydrofolate. So early work with Dihilobacterium showed that methylene tetrahydrofolate was most likely being produced from dichloromethane, and that is methylene tetrahydrofolate is a component of the Wood-Ljungdahl pathway. So once that transformation occurs, then the Wood-Ljungdahl pathway can kind of turn over as we see in other organisms. But the big question is what's kind of dechlorinating the dichloromethane, what's splitting that carbon-chlorine bond. One of the issues with not knowing this as well is that it does somewhat hinder bioremediation applications of this work so far, because knowing that enzyme would be really useful to be able to track it in situ, like I mentioned that we're able to do with the reductive dehalogenase genes. So one of the main goals of this work was to try and identify that initial enzyme carrying out this first step in the pathway. So looking at DCMF, which is the bacterium that I was working with in this enrichment culture, it ferments dichloromethane to acetate in approximately a 2-to-1 molar ratio. And we can see here on the left that after a little brief lag time, dichloromethane is being depleted at the same time that acetate is being formed. And we're seeing a concomitant increase in DCMF cells and also in total bacterial cells, which was measured by qPCR of the 16S RNA gene. We looked as well at the community composition over time during this growth experiment, and that's what's shown in this graph on the right here. So we used Illumina 16ASR RNA gene amplicon sequencing to look at this community composition. And basically what we can see is that when the culture is first inoculated at day 0, we have a relative abundance of DCMF sitting around 55 to 60%. Then during the lag time, the relative abundance of DCMF actually drops way down to below about 10%. And we see a huge increase in these kind of culture, or a huge relative increase in the culture cohabitants. But as soon as dichloromethane starts to be metabolized, DCMF kind of comes back up and is really the only organism that increases in relative abundance relative to substrate depletion. So DCMF is the dominant community member, we would say, particularly during dechlorination, but the composition of the culture does fluctuate in response to substrate consumption. Interestingly, DCMF is also the only one of those 3 DCM fermenting bacteria that is not just an obligate DCM fermenter. So it's also able to utilize the substrates methanol, choline, and glycine betaine. And we saw kind of similar community composition changes in response to substrate consumption when we look at those substrates as well. So we had pretty good clues from the beginning that DCMF was likely the only dechlorinating organism in the culture. And then we wanted to look a bit more closely at how, kind of to confirm that it was using the Wood-Ljungdahl pathway for DCM fermentation and metabolism. So the Wood-Ljungdahl pathway is shown again on the right-hand side here. And essentially what we did was we took the culture and we amended it with 13C labeled DCM. So that's this top left graph here. And the 13C labeled carbon from the dichloromethane at the end of the experiment we found was predominantly found in the methyl group of acetate. So that's showing that the carbon from dichloromethane was being directly incorporated into the acetate that we saw being produced in the culture. And then we kind of did the analogous or the reverse of that. And so we took a culture that we buffered in MOPS medium instead of the typical bicarbonate buffered medium, and we added in exogenous 13C labeled bicarbonate. And that allowed us to see predominantly, again, 13C incorporation from the bicarbonate into the carboxyl group of acetate. So looking at these 2 experiments together showed us that DCMF was assimilating carbon from both dichloromethane and from the carbon dioxide or bicarbonate in the culture. So it is a mixotrophic organism. What we also found was when we looked at the 13C recovery from that first experiment with the labeled dichloromethane, we found the 13C label in biomass, in carbon dioxide, in the bicarbonate that was aqueous, and also within the acetate. And there was actually 128% recovery of that labeled carbon, which showed us that there was also no unknown fate of dichloromethane within the culture. So that was again confirming that DCMF appears to be the only dichloromethane metabolizing organism within this enrichment culture. And basically what we're seeing then in this Wood-Ljungdahl pathway is the enzyme was still unknown, what was transforming dichloromethane, but it looked pretty likely from all the sort of electron balances from this labeled carbon work that it was again being transformed into methylene tetrahydrofolate, similar to what had been found for Dihilobacterium formicoaceticum. And we were then seeing both oxidation to formate onto bicarbonate, as well as reduction into acetate. So we then did some proteomics work. And what was lucky was, as I mentioned, because DCMF is the only dichloromethane fermenting bacterium that is able to use other substrates as well, it gave us this really unique position to be able to carry out comparative proteomics. And so we were able to look at proteins that were significantly different in abundance on various substrates. And that was the first time that this has been able to be done for dichloromethane fermenters. There has been a previous study, proteomics study with Candidatus dichloromethanomonas, but because it can only utilize DCM, it was really looking at sort of starvation and fed conditions, as opposed to the difference in protein abundance, kind of with normal functioning, but various substrates. So again, confirming the involvement of the Wodunga pathway, we saw that it was proteins from that pathway were highly abundant under all growth conditions, as well as those for an ATP synthase, indicating that there might be some sort of chemiosmotic energy generation, as well as like the substrate level phosphorylation going on. But what was really exciting was that there was this cluster of genes that jumped out, and the methyltransferases in this cluster, some of the methyltransferase components in this cluster were among the most highly abundant proteins under cells grown with DCM. When we looked at statistical comparison between dichloromethane and glycine betaine grown cells as well, we saw that these proteins were all significantly more abundant with DCM grown cells compared to glycine betaine grown cells. So you can see here in this heat map essentially that anything as we come up from through the sort of blues to the green and yellow is the much more highly abundant, and that's with a sort of Z-score normalized log2 fold change essentially in protein abundance. And so that's this highlighted cluster here that we think are involved in DCM metabolism. So the 8 genes in this cluster involve a bunch of components for a methyltransferase system, as well as a 2-part regulator and a transporter gene. To briefly cover the methyltransferase components, essentially it's a methyltransferase 1, a methyltransferase 2, and a coronoid protein. And there's kind of a very broad overview schematic of how they work together up here. So the coronoid protein in this is really important, and it's kind of analogous to, we think it could be analogous to the coronoid cofactor that we see in reductive dehalogenase genes that allows that carbon-chlorine bond to be targeted and broken. So we see here that the methyltransferase 1 essentially is known in this sort of generic scenario to have the coronoid protein attached to it and to transfer the methyl group from the compound onto the coronoid protein before a methyltransferase 2 then takes the methyl group from the coronoid protein and transfers it onto the final accepting protein, which is typically tetrahydrofolate. So this kind of system works in demethylation of a whole bunch of various compounds such as methylated amines and methylated glycines. And we're now hypothesizing that similar but distinct biochemistry is occurring to dechlorinate dichloromethane. The one important difference in this is that in these systems, typically it's forming methyl tetrahydrofolate, whereas with DCM, we're pretty certain that methylene tetrahydrofolate is being formed. So there's definitely some interesting biochemistry going on here, and further study is definitely going to be devoted into looking at how that system actually works and kind of confirming biochemically the specific involvement of these methyltransferases in DCM dechlorination. But what was also interesting is that as well as these methyltransferase components, this 2-part regulator, the histidine kinase within that regulator actually contains a POC-R domain, and these domains have previously been shown in the literature to respond to dichloromethane or other small hydrocarbons. So that's another nice clue that this cluster of genes might be involved in dichloromethane dechlorination, and actually transcription of this cluster might be directly regulated in response to the presence of dichloromethane. So the next step was to kind of look and see if we could find any other similar genes or gene clusters within the databases. And another key that this might be involved in DCM dechlorination is that there are actually only 3 close homologs to any of these proteins, and 2 of those are within the other 2 known DCM fermenting organisms. So we can see here that close homologs were found within Dihalobakteri Formicoaceticum and in Candidatus dichloromethanomonas elegantquensis, which are the 2 DCM fermenters. And then curiously, there was also a really close homolog to the whole cluster found within Dihalobakteri UNSW DHB. Its role is way less clear in that. That's actually a chloroform respiring bacterium that our laboratory discovered and has worked with. So it's been shown to not be able to use dichloromethane. This is very interesting, the clusters seen there as well. But what was really cool about this is not only did the proteins have remarkably high percentage amino acid identities to each other, that are all sitting at around like 88 or over 90% amino acid identity, but the actual synteny of the cluster, so the organization of the genes within the cluster, was very highly conserved. Unfortunately for Candidatus dichloromethanomonas, it's not a closed genome yet, so the genome is still present in about 52 different contigs, and that's why we see this split here. So we can't demonstrate that these genes are actually next to each other, but that could hopefully be shown in the future if the genome is closed. It's also worth noting, obviously, that there are these 2 extra genes in the other 3 organisms except for DCMF, and those are actually domain of unknown function proteins. So it's hard to say what their role is at the moment, but clearly DCMF is able to dechlorinate dichloromethane without them. So that remains to be shown what role they have, if any. It is also worth noting that in the proteomic studies with Candidatus dichloromethanomonas elenquensis, methyltransferases shown in this cluster were also amongst the most highly abundant proteins in that work as well. So we're getting pretty good evidence that this cluster has some really important role in GCM dechlorination, and there's just some more work that needs to be done showing exactly how that biochemically works, how that transforms dichloromethane into methylene tetrahydrofolate before it enters the Wood-Lungdal pathway. But there's some nice candidates in here that might be able to be targeted for studies to be able to track in the field when we're looking at dichloromethane fermentation. So to kind of sum up that work briefly with DCMF, this is a metabolic model that we have for the organism at the moment, and I haven't been talking about its use of the other compounds, so you can kind of ignore the top half for the purposes of this talk, but you can see at the bottom that DCM is most likely being demethylated via this putative methyltransferase, transformed into methylene tetrahydrofolate, and then metabolized through the Wood-Lungdal pathway to form acetate. So the other part of this work was essentially this question that I kept coming back to, and my supervisors kept coming back to, which is what are the non-dechlorinating bacteria doing? The culture DFE has been maintained for over 6 years and over 20 transfers. It's always maintained on dichloromethane, which is this, 1 carbon chlorinated substrate that we're pretty certain the other organisms in the culture are not able to utilize. So how are they persisting? Because we tried really hard to isolate this organism. We did a whole lot of series of dilution to extinction transfers, which we hoped would be able to dilute out the cohabitants, but in each case they seem to have stuck around. So the first thing that suggested to me was that there might be something that they're producing that DCMF requires, like some sort of nutrient or amino acid or cofactor, which if we can identify that would help us to isolate DCMF. But it was also just a really interesting question, particularly when you think about sort of applying these cultures within a industrial context. We work a lot with pure cultures in the laboratory, but out there in the environment, pure cultures are very, basically non-existent. So I think it's actually really important to look at, how is this community, which seems relatively stable over a long time period, what is the role of these non-dechlorinating organisms, and are they supporting the dechlorinator actually, or are they hindering its progress? So based on what we know about the identified taxa, we kind of found some information that taxa such as the Synergistaceae and the Spirocutaceae, Disulfovibrio that we were seeing in the community, they'd been associated with hydrocarbon and organohalide degrading cultures for quite some time, but almost never as the actual degrader, except for Disulfovibrio in some cases. And so a lot of people have speculated, and we began to speculate as well, that perhaps they are necromass recycling. So that is essentially taking all of the dead cells and recycling them, recycling components such as proteins, carbohydrates, lipids, and using up all of that sort of dead biomass to persist within the culture. And so we went back and looked at the data that we had already gotten for DCMF, which was a lot of genomic data and a lot of proteomic data. And the first time around when I'd gone through this, it was kind of like, oh, this is so annoying that it's not a pure culture and we're having to fish out the DCMF, the stuff that we care about. And this time it was like, actually, I'm really interested in all the other stuff that's there. So with the help of Dr. Richard Edwards and Xavier Vasquez Campos in the School of Biotechnology and Biomolecular Sciences at UNSW, we were able to pull out all of the other reads from the PacBio sequencing that didn't belong to DCMF and assemble that into the metagenome and bin it into 7 mags and 3 much smaller bins that we couldn't assign taxonomy to. So you can see those here. And then we were also able to use that as a database for the protein searches as well, and identify 328 proteins belonging to these cohabitants. And that was compared to about 1,380 proteins that belong to DCMF from that work. So the majority of the highly abundant proteins from these cohabitant bacteria were mostly for the sort of crucial cell functions that you would expect, such as DNA replication and repair. But when we looked a bit more closely, we could also get some more evidence for this necromass recycling and get kind of an idea as to what the non-dechlorinating community was doing. So we could see, for example, with the spirochetes, that we had 2 spirochete sort of mags, which were also kind of named as treponematales by GT-DBTK taxonomy. And one of those was 92% complete, the other was 43% complete. So obviously a lot more caveats with that second one having a relatively incomplete metagenome or genome. But basically, these kind of organisms were already commonly detected in organohalide contaminated sites. And there was a really nice paper from Dong and colleagues in 2018, which showed that they are bacterial necromass recyclers and they're quite widespread in the environment. What we saw in the mags for these guys was a wide variety of ABC transporters, indicating that it is a versatile scavenger for mono and oligosaccharides, amino acids and peptides, and that they may be able to produce hydrogen, alcohols, and short-chain fatty acids from fermentation of the amino acids. Then when we look at the nitrati disulfovibrio mag, which is essentially just a fancy reclassification of a disulfovibrio species, we found that there were some proteins for an electron transport chain and dissimilatory sulfate reduction, but in the absence of any kind of known sulfate in the culture, in the medium, we think it's actually carrying out a proteolytic function within the DFE enrichment culture, and that's based on some other experiments we did enriching on peptone and essentially getting a pure culture of this nitrati disulfovibrio when it was grown on peptone. Looking at the synergistales, this taxa contains known amino acid fermenters, and so it kind of was fitting that we found a lot of ABC transporters for branched chain amino acids and dicarboxylates, as well as hydrogen-producing hydrogenases. And then the Lentimicrobiaceae are known carbohydrate fermenters, and accordingly this mag was quite rich in carbohydrate active enzymes or cazymes, as well as extracellular proteins, which can be quite useful for breaking down much bigger carbohydrates externally before importing them into the cell. So it might also have a function in kind of breaking down those larger necromass components in order for other bacteria to be able to access them as well. So pulling this all together, we kind of get this big picture view of the culture. And there's a lot more going on there than just the dichloromethane fermentation. So we have DCM being utilized by Candidatus formomonas warabia, DCMF, and producing acetate. But then from all of those dead cells that come from, that biomass turning over, we also see carbohydrates, sugars, amino acids, and peptides being released and being variously utilized by the organisms in this culture. And that was going to produce potentially hydrogen or just shorter chain volatile fatty acids that can then also be reused. So this is kind of exciting because it gives some insight into more community function that's going on. And necromass recycling is shown to make a really important contribution to nutrient cycling in the environment. This is particularly the case in oligotrophic ecosystems, such as these subsurface kind of soil, groundwater environments. Obviously, not as much limited when you have these highly contaminated areas where there's been a lot of carbon come in from the pollutant, but especially as we move towards the plume fringe and it becomes again more oligotrophic, this can be relevant. It's also really interesting because the production of hydrogen and acetate and ethanol can actually stimulate the microbial blooms or serve as secondary substrates for co-metabolism of the pollutant. And the consumption of this dead biomass can remineralize or liberate important nutrients such as nitrogen and phosphorus and trace elements. So that can actually help keep the culture healthy. And for example, there's just one study by Christioletzer et al. That's shown that like a phototroph, Cyanococcus, actually has longer and more robust lifespans when it's co-cultured with this necromass scavenging Rosiobacter species that removes nitrogen-rich wastes. So I think it's worth thinking about these kind of cohabitants and thinking about forming these artificial consortium of bacteria to actually potentially allow us to develop more robust cultures for bioremediation applications in situ. And kind of drawing on that is this idea from Pascual Garcia et al., which is a paper that came out last year, and it kind of defined this term of metabolically cohesive communities, or MECOCOs, which are these subgroups in the environment where the consortium as a whole actually stabilizes the ecosystem dynamics, and it buffers the community against sort of external resource fluctuations via the mutualistic interactions. And the positive feedback loop from this actually makes the metabolically cohesive community, or the MECOCO, far more robust than any of the component species would be individually. So I think that's definitely something worth considering if we're going down the route of bioaugmentation for bioremediation purposes, because there's already going to be a microbial ecosystem present most likely at these sites, and so when we're adding in bacteria for remediation, we want to make sure that it has a good chance of kind of surviving and thriving in that environment to reduce the pollutant levels. It's also worth thinking about, particularly not so much in the case of this DCM fermentation where hydrogen can actually inhibit the fermentation, but when we're looking at organochlorine respiration and it needs hydrogen as an electron donor, that perhaps co-culturing with these necromass recyclers that are producing hydrogen from this stuff can actually provide a constant source of electron donor as well. So that's kind of a nice idea to think about and pursue potentially further as well for more robust cultures for bioremediation. So in conclusion, DCMF transforms dichloromethane to the environmentally benign product of acetate via the Wood-Lundahl pathway, and DCMF cells that were utilizing DCM had a significantly higher abundance of this novel methyltransferase that we propose is involved in carbon-chlorine bond cleavage, particularly because that cluster of methyltransferase genes associated with DCM is highly conserved amongst all three anaerobic DCM-degrading bacteria. And then when we look at the non-dechlorinating community members, they likely persist by necromass recycling, but we're starting to think there could be some really important lessons here and understandings to gain, that they might play an important role in situ. They help us understand carbon and nitrogen cycling in the environment, and it might help us develop more robust cultures for commercial application. And with that, I'd like to acknowledge my supervisors, Mike and Matt, as well as postdoc in our group, Haluk, who helped a lot with genomics of DCMF, Rich and Zabi in Babs at UNSW for their help with the metagenomics and the metaproteomics, and Ling Zong at the BMSF, who helped out with the actual proteomic analysis of this work. And any questions? Thank you.
Sophie, thank you so much for that really interesting talk, fascinating work actually that you did during the PhD. And the necromass recycling story is really interesting. A lot of mileage there to explore in the future, I can see. Now, we've had some questions come through. How I'm going to play this is I'm going to go through the questions and field them to you, Sophie. But those individuals that have asked questions, as I go through them, please turn on your microphones to add to the discussion. I'm going to run this question time for about 10 minutes before I introduce Andy, but at the after Andy's talk, we do have up to 45 minutes for questions and discussions. So if your question hasn't been answered or fielded at this point, doubtful, because I've seen a handful of questions coming through at this stage. So I'm just going to fire off one question from me, Sophie, and that is, do we know where the chlorines are going? So in the dichloromethane degradation, you've tracked the carbon. Do you know where the chlorines are going? Not as specifically, I guess. So generally it's just released as the chloride ion, and that's probably just being excreted from the cell and released into the medium, I would guess. Yeah, but we haven't specifically tracked the chlorine in the culture, no. Yeah, so yeah, I wonder if stable isotope probing with chlorine 35 or an isotope of it would be interesting to see where it goes. But I completely agree. I think it would just be released into the environment, go somewhere. Right, let me begin with Sonia's question. She says, you said the recovery of 13C in the labeling in the first experiment, but I missed the number. What was it?
Oh yeah, so the recovery of 13C from the 13C labeled dichloromethane was 128%. So a little high, but kind of within the sort of typical window of recoveries that you see in these kind of experiments. And that was essentially showing us that there was no unknown fate of dichloromethane. We kind of could see all of the carbon being recovered where we were looking for it.
Okay.
Okay, great. Thank you very much. Really good presentation.
Thank you.
Next question from your previous supervisor there, from Mike here. Sophie, how can you directly implicate the MT1 or MT2 methyltransferases in dechlorination of DCM?
Yeah, so I guess for that we would probably look at doing some enzyme assays, which is what Mike kept pushing me to do in my in the PhD, and I got way more interested in these necromass recycling interactions and teaching myself metaproteogenomics because I got swirled away by the bioinformatics. So yeah, looking at doing enzyme assays would kind of help show us that there's also been a lot of precedent, particularly looking at the reductive dehalogenases with kind of running the protein extract down a Blue Native page gel, and you can then cut out the various bands. And so then you can take each band and do the enzyme assay or an activity assay with that specific band, sequence the ones where you find the activity, and that will give you a much smaller range of proteins, or ideally just 1 or 2 proteins that are much, much, much more highly abundant from that band. And then you'll see, be able to specifically link that protein to the actual activity of dechlorination.
So what do you think about cloning that cluster into another host?
Yeah, cloning would also be an option. Probably into something like an acetobacterium would be useful because it's very likely closely linked in with the Wood-Lungau pathway. So using acetobacterium, which is quite well studied, so we've got sort of the tools for that in terms of the molecular biology of doing the cloning, and it's already got the Wood-Lungau pathway. So you could just try as well cloning the methyltransferase cluster into an acetobacterium or something similar as well.
Yeah, cool, thanks.
And here, are we talking about isolated strain that you have, or non-isolated, cloning of non-isolated organisms?
So yeah, our organism isn't isolated as yet. It's still present in the enrichment culture, but yeah, I think we would be able to potentially ideally be able to clone the methyltransferases somehow. Yeah.
Okay. It'd be really, really cool if eventually you might be able to start purifying these enzymes if you can go that far to determine Km values and Vmax and that sort of thing.
Yeah, that would be very ideal.
A question here from Louise. What do you think are the rate limiting nutrients for these communities? Cobalt as an example?
Mm-hmm. Yeah, definitely cobalt and cobalamin, probably, to get that cofactor that's required, we think is required, at least for the methyltransferase. Mm-hmm. That's kind of the main one, and we think that, we do add exogenous vitamin B12, so cyanocobalamin, to the cultures, but it does look like from the genome that DCMF is also able to produce its own cobalamin cofactors as well, so it's kind of equipped to do its own thing out in the environment. Yeah, so cobalt and cobalamin would be pretty key.
Right. Question here from Terry McGinty, down at Essex. Really interesting presentation. What was there an absolute increase in numbers after addition of DCM, but before degradation? And he also asked, why is there a relative decrease in DCMF? And what is the source of the necromass before degradation of DCM before DCM starts? Did you want me to start with the first question there, Sophie? You got them all?
I think I got it all. So if we go, I'll just go back to the DCM graph. So yeah, unfortunately, I didn't take as many samples for the qPCR for the quantification sort of in that lag period. But you can kind of see that before DCM dechlorination has really kicked off, there is still an increase in both the total bacteria, which is the white squares, as well as a little increase in DCMF as well. So I think it's possible that the relative abundance shifts because there is actually a real increase in the number of the Synergistaceae in this case, as well as the Disulfur Vibrio. And there might be just enough sort of senesced biomass in the inoculum that they're able to kind of kick off and start producing. But essentially, yeah, we don't see any real like turbidity in the cultures just by visual assessment. The cultures don't start to look turbid until DCM dechlorination really kicks off. But yeah, I think there could be like a small amount of growth that's happening from the necromass fermenters there. There might also be, I think, just some of the DCMF cells kind of dying essentially, which is why we're seeing this decrease in relative abundance in the lag time, that they might just be kind of, the shock of the transfer and adjusting to the new medium as well before stuff kicks off and they kind of regain their footing.
Okay.
Thanks, Sophie. That makes perfect sense. I'm sorry, I missed the qPCR data.
Yeah, thanks.
Thanks, Terry. I'm just looking through the chat line here. There's a comment here by Sonia Heaven. I'm not active in this area, let alone an expert, but fully agree that the necromass recycling aspect is likely to be of very broad interest. If the presentation is made available, I'd like to recommend it to some colleagues. Yeah, we'll certainly make that presentation. Is that okay with you, Sophie, to have this presentation available, or is there any sensitive information there?
I will double check with my supervisors. So the necromass recycling stuff is kind of coming out in a paper that we are about to submit. So it's not quite widely available yet, but I can also let you know there should be a preprint online within the next month once we submit it. So that can be, yeah, definitely happy to share it once that's happened.
Great. If you're okay to allow us to if you can send us your presentation, but that's only if you want to, and if you do, you can cut out some slides or some information if there's anything that you prefer to keep under wraps.
Yeah, sure thing.
Let's see. Yeah, one more comment here. Let's see, from Matthias Beck. Would it be viable to use DCMF in an industrial anaerobic plant for bioremediation, maybe with sustainable produced hydrogen gas addition if needed, maybe comparable to processes as used by electrochea producing methane, or is something like this already done?
So DCMF, I don't think we have used commercially yet, but it's definitely would be plausible within within an industrial capacity in like an industrial bioreactor or something. It wouldn't need any exogenous hydrogen. That hydrogen actually inhibits the fermentation. So it's pretty self-sufficient in that regard, not being an anaerobic respiration process. Yeah, so it would be pretty cool to get it out there and see it do its thing to help remediate other sites as well.
Any other comment, Matthias? Or
no, thank you very much. Thank you very much. That's very great.
And various other comments there through the chat line, thanking you for an interesting talk there, Sophie. Thank you so much. That was absolutely brilliant work.
Cool. Thanks so much.
I know we'll have a discussion later after Andy's talk. Question times for him as well, and then we'll have a discussion. But I just wanted to mention, are you still looking for a postdoc, Sophie? Because this is a great opportunity to plug that.
I am still shamelessly self-promoting and looking for a postdoc. Yes. Yeah.
There'll be a couple of postdoc opportunities in my lab. I'll send you information by email about that.
Thank you. That would be great.
All right. So we'll move on to Andy's talk without further ado. So it gives me, so as I mentioned at the beginning here, that the biographies of the speakers were made available during advertisement of their talks. So I won't take up time of the speakers by introducing them with a biography. So without further ado, I welcome Professor Andy Cundy to begin his talk entitled Integrated Attenuation Strategies for Problem Organohalides.
Thanks, Tony, and hello all. I'm just sharing my screen, so hopefully this will work okay. Okay, can you all see that okay?
That's perfect, Andy. That's right.
Right, let me go back to the first slide, sorry. Okay, well, first of all, I'd like to thank Tony, Angie and Louise for organising this seminar and for inviting me here today. I'd like to thank you all for attending virtually. It's nice to meet you all online. My presentation today is all about integrated attenuation strategies for problem organohalides. So as Tony's mentioned, I'm Professor Andy Cundy and I'm based at the University of Southampton. I'm also affiliated with the Hong Kong branch of the Southern Marine Engineering Laboratory at Guangzhou, where I do some joint work in the Pearl River Delta, which I'll talk about in a little bit more detail later. Okay, so content of the talk today. First of all, I'd like to introduce organohalide contaminants generally, following on from Sophie's presentation. And then I'd like to talk a little bit about integrated attenuation strategies. What are they and what do we mean by this term? Then I'd like to give you two examples. The first one on tetrachloromethane or carbon tetrachloride. looking at green remediation of carbon tetrachloride with wider benefits based on some joint work we've been doing with the Argonne National Laboratory in the US, essentially looking at integrated remediation strategies for quite a large scale carbon tetrachloride contamination problem they've got in the US. So example 2 is on PFAS, perfluoroalkylated substances, and other emerging contaminants in coastal brownfields. And what we've been looking at there are their interaction with sediments. So I'm going to present some recent field data we just published in the Journal of Water Research. And then I'd like to talk in a little bit more detail about an EdNet funded project by the BBSRC, UKRI BBSRC in the UK, looking at enhancing the interaction of PFAS with sediments and enhanced natural attenuation strategies. So organohalide contaminants, as I'm sure many of you know, these are a very wide range of often persistent, mobile and largely recalcitrant contaminants. So they're organic compounds containing one or more substituted halogen atoms. And what I'm going to focus on today are tetrachloromethane or carbon tetrachloride, traditionally used in propellants and refrigerants as a dry cleaning agent and also as a fire extinguisher. It's been used in making nylon, as a solvent for rubber cement, soaps, insecticides, etc. So very wide industrial usage. And PFAS, these are key emerging contaminants which have been used for over 60 years in a wide range of industrial applications. So for example, firefighting foams in adhesives, as waxes, in non-stick cookware, etc. With the result, they are now pretty ubiquitous in environmental matrices, and it's worth flagging these are the focus of a great deal of ongoing environmental and also remediation research. Okay, so what are integrated attenuation strategies? Well, when I talk about integrated attenuation strategies, essentially these are strategies that utilize natural attenuation reactions enhanced by soil additives, plants, barrier technologies, soft engineering such as constructed wetlands, etc., to protect receptors by removing or stabilizing the source of the contamination or breaking the contaminant pathway or linkage. So in the diagram there, we've got the generic source pathway receptor model that's used for contaminated land management quite widely. So that's termed a contaminant linkage. So we have a source and a receptor and a pathway by which the contaminants from that source reach the receptor. If you've got all three components, you've got a contaminant linkage and you've got an environmental risk. So if we're going to minimize that environmental risk, we either need to, in order to protect the receptor, we either need to remove the source or break the pathway in some way. And this is what natural attenuation strategies, monitored natural attenuation or enhanced natural attenuation strategies intend to do. The rationale behind this is that these strategies use natural degradation reactions or natural removal reactions such as biodegradation, sorption, chemical reactions, dilution and volatilization to reduce the mass flux of contaminant moving from the source and reaching the receptor. So example there in the lower part of the slide, we've got a dissolved plume coming from a source. And essentially here, these various reactions reduce the mass flux sufficiently so that the receptor is protected. Okay, so by the time the mass of contaminant reaches the receptor, it's below regulatory or threshold guidance. And when we talk about enhanced attenuation, basically we're intervening to increase the rate of degradation, to increase sorption, or to increase volatilization, etc. So these are integrated approaches, and importantly, if they're effectively designed, you can use them to realize a range of wider social, economic and environmental benefits. Now, we first started working on this area a few years back in European Union Framework 7 program called Greenland. Okay, it wasn't based in Greenland, but it was all about greening land for remediation purposes. And here we were working on what we term GROW, gentle remediation options. These were risk management strategies or technologies that result in a net gain or at least no gross reduction in soil function. So they're preserving the soil function while at the same time achieving effective risk management. So particularly in Greenland, we were looking at phytoremediation, plant-based management strategies where you use plants and their associated root microbiome to break down contaminants. So the general concept here was that phytoremediation and other gentle remediation options can be applied as part of wider risk and land management strategies. So you apply them either alone or in combination with other more intervention-based remediation techniques to gradually mitigate or eliminate contaminant-induced health and environmental risks. Importantly, while simultaneously realising a range of wider sustainability benefits, economic, ecosystem, service and other benefits. So as the diagram below shows here, we're using plants to either gradually remove the source term by phytoextracting contaminants or by phytodegradation, or we're using them to break the contaminant linkage in order to protect the receptor. So what are these wider benefits? Well, we published this diagram in 2016 as part of a wider review of phytomanagement, but here we can model along the source pathway receptor, showing that plants are capable of gradual removal or immobilization of the source term by phytoextraction. They can also reduce the labile pool, that mobilized pool of contaminants, leading to a rapid reduction in flux of contaminants to receptors that may be at significant risk. And also you can use vegetation to manage receptor access to the subsurface by providing a green cover. In doing so, you can generate a wide range of additional benefits. So leisure and recreation benefits if you're creating urban parkland, if you're growing biomass for economic return, obviously you can generate economic returns to the local community. You can enhance ecosystem services, allow flood protection by providing green cover on a site, health benefits, and also carbon sequestration. So this model, this kind of phytomanagement model, places realization of wider benefits of the core of site design. So here, plant-based technologies and other gentle remediation technologies, including bioremediation, can be applied as part of integrated mixed site risk management solutions. Or equally, you can apply them as part of holding strategies for vacant sites, or to provide economic leveraging for site redevelopment, if you're going to redevelop that land as urban parkland, or for biomass returns, etc. So coming on to the first example, tetrachloromethane, green remediation with wider benefits. This has recently been published in Journal of Cleaner Production, so hopefully that's going to be online in the next couple of weeks. The preprint is available online. So this was an integrated system example, and this was looking at a carbon tetrachloride contaminated site in Nebraska in the Midwest USA. So a schematic of the integrated remediation screen there, this scheme is shown on the right of the slide. And this was an integrated remediation system. So the problem here was that carbon tetrachloride had been used as part of a fumigant, essentially as an insecticide on a former grain storage area, a former US Department of Agriculture facility. So it was used, a carbon disulfide and carbon tetrachloride mix, carbon tetrachloride is the main contaminant of concern here, was used as an insecticide on the grain store and found its way into groundwater. Okay, and the groundwater migrated northwards until it reached a local creek, and it was found that in that creek there were significant values of significant concentrations of carbon tetrachloride, significantly exceeding the federal standard of 5 micrograms per litre. So Argonne National Laboratory, in combination with the US Department of Agriculture and also a local community, implemented this mixed remediation system. Okay, so nearest to the source of contamination, an air stripping scheme was implemented, essentially pumping of contaminated groundwater to air for irrigation of an athletics field and volatilization of carbon tetrachloride to air. Element 2 and element 3, though, are what I'm going to focus on here today. So element 2 was a phytoremediation zone, and element 3, constructed wetlands, which were implemented as a final polishing stage to clean up any carbon tetrachloride that escaped element 1, the air stripping, and element 2, the phytoremediation component. So the overall aim here was to permanently reduce carbon tetrachloride concentrations in the site groundwater and protect down gradient surface waters. So a greater than 10-year monitoring program was carried out on surface water, groundwater, air and vegetation samples led by the Argonne National Laboratory. So trees were initially planted in 2005, And following that to the present day, there has been at least annual sampling of surface water from various locations, ambient air, groundwater samples, and also vegetation, tree tissue, leaf and grass samples. So the basic philosophy behind the phytoremediation component of the integrated remediation scheme was to encourage deep-rooted trees to access the contaminated aquifer. And then the main mechanism of carbon tetrachloride removal was predicted to be uptake by plant roots, followed by translocation and dispersion to air, essentially a hydraulic pumping system. Or degradation within root or above ground plant tissues. Okay, it's important to note here that groundwater here is aerobic, dissolved oxygen concentrations in groundwater here between 2 and 10 milligrams per litre. It's important to note also that between the trees, native grasses were planted. So this is basically an area of prairie. So the idea here was to plant native grasses and wildflowers to actually increase the biodiversity of the area, but also to intercept any falling rainwater before it reached the subsurface. So the idea here was that the tree roots would primarily draw water from the aquifer rather than from infiltrated water from the surface. So why was the hydraulic pumping thought to be the dominant mechanism here? Well, carbon tetrachloride uptake by vegetation is favoured in oxic subsurface conditions, as we find at the Murdoch site, the site I'm describing, over in situ biodegradation reactions. Now, in the groundwater samples that were tested, chloroform was detected at less than 1% carbon tetrachloride, and methylene chloride, a degradation product of chloroform via reductive dechlorination, was not detected in groundwater samples. However, there was some evidence for partial carbon tetrachloride degradation, either within plant tissues or in the root zone prior to uptake. Based on sacrificial tree sampling, there was a positive correlation, as shown in the diagram here, between chloroform and carbon tetrachloride in branch tissue samples. So there did seem to be some breakdown going on within the trees or in the root zone prior to uptake. So was the system a success? Well, there was an average reduction in carbon tetrachloride concentration across the phytoremediation zone of about 99% over the period 2006 to 2012. So basically here we've got data from 3 surface water monitoring points, SWM, and you can see at the beginning of the implementation of the scheme quite high carbon tetrachloride concentrations. Bear in mind the federal standard is 5 micrograms per litre. Particularly in site SWM1 at the entrance to the phytoremediation zone. By the time we exit the phytoremediation zone at SWM2, you can see that obviously the concentrations there are much lower, they're variable but much lower. And then at SWM3, which is the final polishing stage, the constructed wetland stage, we are below the regulatory standard. Okay, so effective protection of the compliance point. And it's estimated that between 300 and 600 grams of carbon tetrachloride were removed annually by the phytoremediation component. So here we see an effective risk management with wider benefits, and that's an important point. And this is what the paper we've just recently published focuses on. So carbon sequestration benefits. An estimated 77 tonnes of carbon dioxide sequestered per hectare per year. That's about 120 tonnes, in excess of 120 tonnes total per year. Increased floral species richness. There was increased community participation and education benefits. This is used by the local community as an outdoor classroom. There's also flood protection benefits as well. Okay, so the second example. PFAS and emerging contaminants in coastal brownfields. What I'd like to look at here is some work we've done on interaction with sediments based on field data in the Southampton water area on the south coast of the UK. So if we think about sediments, estuarine sediments and contaminants, we know that estuarine sediments can filter and trap a wide range of contaminants. So there's an example of some data we published a few years ago looking at the retention of radionuclide and heavy metal profiles within estuarine sediments here within salt marsh sediments. So as you can clearly see from the diagrams there, from the graphs, these show in the case of cesium-137 activity versus depth, in the case of copper concentration versus depth. And we can see a clear subsurface maximum retained in 1992 and 2014 at the same sites for cesium-137 and also for copper. The cesium-137 subsurface maximum relates to fallout cesium-137 from global nuclear weapons testing around about 1963, so in the early 1960s. The copper maximum relates to discharges from the Fawley oil refinery, which is located in Southampton water. Copper was used as part of a sweetening or cracking process in the hydrocarbons, and that was discharged in the late 1960s, dominantly before effluent cleanup measures were implemented in 1970. So we get a clear pulse of copper retained in the sediment dating to about 1970. So these sediments show retention of input maxima related to fallout and local industrial discharges. But the question is, how effective are estuarine sinks for organohalide and other emerging contaminants? Well, in recent work, we've examined 7 emerging contaminants, including pharmaceuticals, plasticizer, but also PFAS, in dated intertidal sediment cores from 2 contrasting systems: Southampton water, which is shown in the upper part of that Google image, which is heavily urbanized and industrialized, and the other, which you can see just at the bottom there to the right of the Google word, is the Bewley estuary. Okay, so this is in the New Forest National Park. It's a much less theoretically contaminated area, much more rural, very non-urbanized. And we've just published this in Water Research. Okay, so it just came out. It's available online over the past few weeks. So the main concentrations, interestingly, of each analyte within the sediments sampled from Bewley and Hythe salt marshes are similar. So we're interested in particularly here on PFASs, and so we can see PFBS, PFNA and PFOA here. Interestingly, the mean concentrations are similar between Hythe and Bewley. And also, if we look at radiometric dating of the sediment cores, so we dated these sediment cores and we looked at the distribution with depth of a range of these contaminants, we found that emerging contaminants, including PFAS, were present at depths which predate their widespread environmental introduction. So the ECs are apparently relatively mobile, including PFAS, in the marsh systems studied. Subsurface concentration maxima relate to local flushing or diffusive processes, and they can't clearly be linked to likely input trends or changes in sediment geochemistry, including sedimentary organic carbon content. So we don't see clear peaks like we see for copper and cesium-137. These are much more mobile. And also, the similar concentrations between Hythe and Bewley indicate there's not one single dominant source in that system. These are fairly ubiquitous. There's a wide range of sources within both systems, and these are being mixed around by tidal circulation before deposition in the marsh systems. So the estuarine filter here shows reduced potential to sequester the 7 emerging contaminants examined and mediate their supply to coastal and shelf environments. The question we're asking now is this consistent at other sites? Well, in a joint project with the Hong Kong University of Science and Technology, this is what we're currently examining, the Pearl River estuary. Obviously a much larger, very different system, which has been subject to very rapid recent industrialization. And here we're looking at sources, fluxes, including historical changes in pollutant loading, and the extent of sedimentary trapping or degradation for a range of emerging contaminants. And also importantly, interventions where we might be able to reduce some of those fluxes going into the marine environment. So key point for today's presentation is also, we've observed that these contaminants are present in the environment. Okay, there's some trapping, but they're fairly mobile. Can we enhance trapping and breakdown of these contaminants in coastal brownfields, but also in estuarine salt marsh sediments? Well, if we think about brownfields more generally, we did some work a couple of years ago in Colombia looking at biochar and soil amendments. And I know that a number of the people on this call have done a lot of work recently and in the past on biochar amendments and how they can be used to promote sorption and also biodegradation of contaminants. So here we looked at artisanal gold mining sites in Colombia, which are grossly contaminated with mercury. And we worked with R3 Environmental, Paul Barlas's team there, and also Secure Solutions, who are commercial providers of modified biochars. The aim here was to look at whether we could stabilise mercury in the soil with soil amendments, with biochar, and then establish a vegetative cap of green cover or biomass for environmental and health protection, but also providing a potential local income source, so non-food biomass. So could you produce usable biomass products on these sites, but also you're also getting carbon sequestration and other benefits. So the work, it was very preliminary work, but it suggested that even with the addition of less than 1% amendments by mass to the soil, biochar, we were capable of reducing the amount of water soluble mercury quite significant. So the water extractable mercury concentrations were significantly reduced following amendment application. So what we're doing currently in a new EBNet project, working with Fred Coulon and others at Cranfield, Exeter and the University of Brighton, is examining whether we can use this kind of integrated approach to enhance interaction and enhance natural attenuation of PFASs. So here we're looking at attenuation of PFASs and or their degradation products by coastal brownfield and salt marsh or wetland soils and sediments, through looking at sorption and degradation, and also comparing biotic and abiotic processes on bulk soils and sediments, but also enhancing natural system attenuation by addition of soil amendments and biostimulants. So here we're looking at granular zero-valent iron. Zero-valent iron has been shown in its nano form to be able basically to crack open PFASs and make them more amenable to subsequent biodegradation. And also we're looking at biochar, both as sorbents but also as hosts for microbial communities that might have some success in breaking down certain PFASs. And we're looking at integrating these in various possible source and pathway interventions. So both tackling the source but also breaking the pathway through interventions such as permeability barrier systems, etc. So we're looking at integrative in situ gentle enhanced bioremediation approaches, which aim to protect local estuarine ecosystems, but also importantly circumvent the need for more intrusive physical and chemical remediation methods such as dig and dump, ex situ stabilisation or ex situ thermal treatment. These may limit subsequent land reuse. So the overall scheme and model here is trying to develop enhanced natural attenuation strategies to protect the receptor by source removal, stabilisation, degradation or pathway management with wider benefits. And this is an idea we've presented in a recent paper in Frontiers in Environmental Science. And this is where we're promoting this idea of actually gentle remediation options, gentler interventions to actually promote enhanced attenuation strategies and realise wider benefit. An example that we worked on in the past is the Port Sunlight Riverside Park in the Mersey Estuary. You can see some pictures there of that site. This was a coastal landfill site that is managed by a group called the Land Trust as urban parkland. And as part of that, we've been looking at how we can quantify both economically and through wider benefits the economic, social and environmental benefits provided by sites such as this. There are various ecosystem benefits, leisure benefits, and also benefits regarding integration of marginalised members of the community. This site is managed by local individuals with autism and their families. So obviously much wider community benefits available there. Okay, so I'd like to finish that very broad ranging talk, and I would welcome any questions. Just to know, acknowledge a range of co-authors there, particularly Paul Bardosh, Yanu Lee, Lorraine Lafreniere, Omar Celis Hernandez and others. Note Fred, Martin, Rich are working together with me on the EBNet project, and we're also collaborating more broadly with Tony on this work also, and various funders, notably BBSRC via the EBNet network. Thank you all.
Thank you, Andy, for that brilliant talk. Oh, I'm getting a bit of an echo here. I hope you're not getting double my voice. Anyway, so I'm just, I welcome all who are attending to just post questions and comments on the chat line. So instead of just reserving this session for questions to your session here, your talk, Andy. I also welcome further questions and comments to Sophie's talk. So I think we just bring in the conversation and discussion for both speakers. Now, I just want to kick off, Andy, with somewhat a bit of a general question here. I mean, what do you find as the biggest challenge in the reclamation of sites contaminated with PFAS? And also, what environmental sites are the most challenging? Are they marshes? Are they coastal? Are they more inland? What are the most challenging?
Really good questions, Tony. And I think that one of the major challenges with these sites is actually really the lack of effective rapid remediation technologies. I mean, there are various commercial products available now for PFAS management, particularly injection of materials into groundwater to essentially treat contaminant plumes. I guess one of the big challenges is around source treatment. You know, the pathway management. We're looking at lower concentrations, of course, of PFAS. We're looking potentially at some pre-degraded products. I think source management is a key issue there. And in terms of problem environments, I think these are really ubiquitous contaminants. We've deliberately focused on coastal brownfields because we've got some really tough environmental gradients there, going from basic terrestrial to marine environments, very contrasting systems, and some really sensitive marine ecosystems. And I think the work at Southampton Water and Bewley has shown really, and this is supported by other work elsewhere, the ubiquity of these contaminants.
So Andy, are they found in polar regions that far, like Antarctica or the Arctic?
From my understanding, Tony, I think they're pretty globally ubiquitous. I mean, they're so mobile and recalcitrant. You know, they're pretty much everywhere, and I think that's a key issue. You know, that even the work we've done on a very limited scale in Southampton Water and Bewley, you know, you're not picking out key individual sources. You're not able to say, right, it's from this particular factory or this particular site. I mean, that makes remediation, you know, much and management much harder. You know, because if you can't fingerprint a particular site, it becomes much more difficult then.
Yes, yeah. I had more questions in mind, but I've got questions coming through in the chat line, so I better step back here. So I've got a question here for you from Terry McGinty, who says, would you please comment on the main sources of PFAS that you will be focusing on?
Yeah, good question. Initially, we're going to be focusing on PFAS released as part of fire training activities from firefighting foams. So essentially, there is a local site. I can't reveal its whereabouts, but there are issues around former firefighting activities there. This is quite a common source for PFAS contaminants, quite a common point source. Which makes it a good kind of first step, I think, for looking at integrated remediation strategies before we move on to kind of more diverse sources or diffuse sources.
Thanks, Randy.
So as I ask these questions, please, I welcome the person that has asked these questions as I'm asking them to turn on their microphones and video if you like to come in on the discussion. However, if you feel that the questions have been answered and you prefer not to, that's completely fine as well. A question here from Louise Byfield. Do you find PFAS contaminated sites have lots of other contaminants? How does that impact achieving the overall site cleanup goals?
Great question. Answer to the first part, yes. There are other co-contaminants at sites. I mean, it is site dependent. And I think, you know, in some sites the PFAS are the key contaminant of concern. At other sites there will be more secondary contaminants. That does complicate the overall site cleanup goals. And I think this is one of the important points to make really with these integrated remediation strategies. What we're looking at really is making them suitable for a range of contaminants. So particular focus on mixed contaminant plumes. But that will be site specific. You'll have to engineer that in a site specific way, because what you don't want is one intervention for one contaminant that then triggers problems, possibly with breakdown products of another contaminant.
Thank you for that.
Related to that question, I thought I'd just plug in with one. I mean, there are a whole different types of molecular weight structures, right? Range of PFAS. Is it known what types are can you rank them in terms of worse to less toxic or less, you know, problematic? Has anything been done in this?
It's a really interesting point, and I think to be honest, PFAS are, as we know, are an area that are just really beginning to be the subject of massive international research. So, you know, if you ask me that in a year's time, I'll probably say yes. I think at the moment it's still under a lot of research. What I would say, one of the interesting things we found with the environmental work in Southampton Water, looking at mobility, the PFAS as we looked at have got, if you look in terms of the octanol water partition coefficient, I mean, it's not an ideal measure, excuse me, but these PFASs have a range of log KOW values from about 1.8 to 7.3, and so you would expect at the higher end of that for them to basically interact quite strongly with particular organic matter in salt marsh systems. They seem to be reasonably mobile, actually, so you know, that's quite interesting that they are, based on predictive models of their, you know, of their absorption, they seem to be more mobile than we'd have expected.
So here you've mentioned about mobility, but that also, and sorption, but that also translates a bit into solubility, right? So there'd be a range of solubilities here for these different PFAS chemicals. Yeah,
correct.
The next question comes from Judith Hagen, and please, Judith, turn on your microphone if you to come into the conversation if you wish. But I also welcome Sophie Holland to come into the conversation if she wishes, because I think this question sort of relates also to her. The question is, are people looking at microbial solutions for PFAS contaminated sites? I have a question that I'd like to join to that, and that is, are there microorganisms that can degrade PFAS but also have a role in degrading chlorinated or organochlorines as well? So are there organisms that can sort of do both? But let's go back to the first question. Are people looking at microbial solutions for PFAS contaminated sites?
I don't mind starting off on that one. Again, great question. The answer is yes. Obviously, there's been, well, Tony and I are aware of some work we've been discussing, which was performed in the US, looking at Acidimicrobium bacterium A6. This was published in Environmental Science and Technology last year, which basically in laboratory-based batch studies, there's evidence that that's capable of breaking down PFAS. That's a microorganism that thrives in low oxygen conditions and is present in the natural environment. So I think people are looking at this now. Whether or not the microbial solutions will be sufficient on their own, this is what we're thinking about with these integrated remediation strategies. Do you need to apply soil amendments, or do you need to apply some of the technology that can actually crack open the PFAS and make it, or cleave it and then make it more amenable to bacterial action? I think that's still an outstanding question. There is a lot of work being done at lab scale. I think that needs to, of course, move to field scale, really, or looking at real contaminated complex materials.
Yeah. So do we know if there are microbes that can do both organohalides and PFAS? Is there any information on that? Or are microbes really focused on, I can degrade PFAS, but I won't touch the organochlorines?
That's a very good question.
And I welcome Sophie to come in on that if she wishes.
Yeah, I mean, I think just to start off, I'll introduce Sophie, but I think the Acidimicrobium bacterium strain, I think, is capable of, you know, it's not PFAS specific. It will break down other contaminants as well. But Sophie, we'll welcome your input on that.
Yeah, I mean, I think it sounds like Andy's a lot more across it than I am. I know that, yeah, like Andy mentioned, there's work being done on bacterial solutions for PFAS contamination, but my kind of very basic understanding is that because they're just such complex molecules and so unlike anything that you find in nature, it's been really difficult to find microbes that can sort of act on PFAS molecules. Like when you look at a lot of the other organohalides and organochlorines, they have sometimes a small percentage that's naturally occurring. So there's been this chance for like evolution over a much longer time scale to evolve these reductive dehalogenases and breakdown pathways, whereas PFAS is so unlike stuff that was already in the environment that there's been this really much shorter time period for microbes to evolve and adapt to these molecules. So it's probably a good place to start with the organohalide, the known sort of organohalide degrading bacteria, but there might be other solutions as well and other microbes out there that surprise us.
Yeah. Thanks, Sophie.
I think just to add to that, Tony and Sophie, I mean, I'm aware of a number of projects that have gone forward for funding in the US in particular, and they tend to be focused more on sorption, using injected carbons, etc., as sorbents. But they're also, as a component of that, looking at mixed microbial consortia, just to test to see whether there's, there is real potential for the PFAS breakdown, or whether actually these injected carbons can act as hosts and substrates for bacterial action that might be able to break down the PFAS. But the focus is dominantly on sorption, but I think there is a lot of potential to explore areas for biodegradation.
Thanks, Andy. Thanks, Sophie. I welcome the community to plug in their questions on the chat line there, please, because it seems like the last question posed there was by Judith. So please, if I don't see any further questions in the next minute or two, we'll bring this session to an end. Meanwhile, I have another question. We always talk about bacteria, and bacteria are really key organisms when it comes to removal of pollutants from the environment. I know with hydrocarbon degradation, the archaea have come to being recognized as also being involved in hydrocarbon degradation, only just in recent years. Never has this been known before. It was always the bacteria as the protagonists, and they are the protagonists. But now archaea do play a role. Do the archaea play much of a role in PFAS or organochlorine degradation?
I can jump in and just say I'm not sure. Not something I've really looked into, but I haven't come across sort of any papers that describe a role as much. I think, yeah, with the sort of organochlorine stuff. I think specifically looking at the only sort of archaeal interaction that I've looked at is more just with methanogens, typically actually inhibited by stuff like the chlorinated methanes and dichloromethane, just because they're sort of methane analog compounds. So yeah, I haven't heard of it for something with chlorinated methanes, but happy to be disagreed with.
Yeah, no, science is that pernickety field where a new discovery just pops up that you never knew of, and like with the discovery of the archaea just very recently, that they can degrade hydrocarbons, and another paper came out not long ago. We had always known the genus Cycloclasticus to break down aromatic hydrocarbons. Then there's been a recent discovery that they can actually break down alkanes as well. So yeah, there's who knows in the next few years whether a new archaeon. In your SIP work, Sophie, did you only look at 16S for bacteria, or did you also use primers to detect archaea that might be incorporating the 13C from the organochlorine?
So we were only using bacterial primers, but that's because the enrichment culture that we have is only bacterial at this point. So it actually was initially methanogenic culture, but we used BES and eliminated them from the culture. So we were just looking at bacteria. But I think a colleague of mine who just recently submitted her PhD thesis looked a lot more broadly for sort of DCM degrading genes within the environment, looking at sort of sludge samples from wastewater treatment plants, and she was able to find a bit of a wider range of this sort of like dehalogenase genes and different organisms that might be involved in the breakdown of DCM, both aerobically and anaerobically.
Okay. Yeah, that would be really interesting to explore that. Yeah. So yeah, I've been doing a lot of SIP experiments in well, a lot. I've been doing some SIP experiments in the past few years, and I've always focused on bacterial, but I've got my students now, postdocs, to actually include archaeal 16S genes to detect for any incorporation of the labeled carbon, just in case we pick up anything interesting.
Yeah, it's definitely worth doing, especially with, like you say, for those sort of surprise discoveries that you might have overlooked otherwise.
So I've got a question here that's coming from Alejandro More Abelanda, and the question is, please elaborate more on the sorption strategy. I think that might be for you, Andy.
Yeah, happy to take that one again. Great question. And I think the sorption of PFAS is an area that's still an area of active research. Certainly, there was a really interesting paper published in 2019 in Environmental Science and Technology, which talked a lot about the sorption mechanisms in soils. And some of the PFAS can actually, depending on the environmental conditions, can be cationic in form, and so they may interact with inorganic soil components as well as organic components. And I think there's arguments that there are electrostatic interactions, hydrophobic interactions as well. I think it's largely going to be site and environment specific. So it's not as simple as or straightforward as for some other environmental contaminants, where the sorption is quite straightforward and consistent depending on environment. Here it seems to be very, very complex, and it's still an area of ongoing research. And I think one of the key points on the sorption is how irreversible it is, because of course what you don't want is a plume management technique where essentially you lock away your PFAS but only temporarily. And it gradually leaks out over the following weeks. So again, I think this is one of the areas we're going to be tackling as part of the EBNet project, looking at exactly how the sorption occurs onto salt marsh soils and also terrestrial soils as well, and whether that truly is environment specific or whether you can pull out more general trends.
Hello, can you hear me?
Yes.
So how do you remove the material later on? If you apply, for instance, a biochar, you use biochar for this sorption purpose, how do you remove it later?
The biochar, it depends on the method you use to apply it. If you apply it as a soil amendment, then basically it needs to stay in situ. So you would need to ensure that that sorption was irreversible, or you were generating biodegradative conditions, whereas you're gradually breaking down the PFAS so they sorb, and then you degrade them. If it's in a permeable reactive barrier type scenario, you could theoretically remove the material and replace it, and then basically incinerate the biochar. So I think it depends very much on the method of application that you're looking at. There are obviously a range of methods you can use for applying biochars.
Right. If it's degradable, it's not really an absorption strategy. It's like you provide a substrate for the microbes to be able to accelerate their growth, and in that growth they eat as well your PFAS. It's not really absorption strategy, in my opinion.
Yeah, no, I think it's a combined sorption and biodegradation. You're practically, I think, in the environment see a combined sorption and biodegradation strategy. And what we're trying to work out is whether which is a dominant component of that. Is it dominantly sorption, or is it dominantly degradation? I think it would be better if it was dominantly biodegradation, because then you haven't got to worry about reversibility of sorption over time. But I think you'll see both processes interacting in the environment.
Okay, very informative. Thank you.
Great. So I'm not seeing any further questions coming in on the chat line. I think it's probably time to bring this session to a close. Before I do pass it on to Angie or Louise to close the webinar, I just want to thank both speakers for your time. Those were two brilliant talks. Hope to have you again sometime in the future. We'll see. But thank you so much again to Sophie. Look, I think it's 9:30 in the evening at night there. Is that right?
Yeah, it is.
So thanks so much for taking your evening to give us this talk for this webinar. That's really very much appreciated. Yourself also, Andy, 9 a.m. in the morning, but I guess that's pretty typical for us.
Thanks, Jeff.
And thank you all who have attended this webinar.